Thanks. We're going to move on now to our contributed speaker today, which is going to be by Dr. Emiliano Altamura, who is a researcher at the University of Bari in the chemistry department.
Hello.
Thank you, Dr. Altamura.
You're welcome. Can you see the screen?
Yes.
Okay. So thank you, Jacqueline, for the introduction. I'm Emiliano Altamura, a researcher at the chemistry department of the University of Bari. And first of all, I would like to thank the organizers for giving me the opportunity to talk to you about my work entitled Light-Driven ATP Production Promotes RNA Biosynthesis Inside Hybrid Multi-Compartment Artificial Protocells. Just for geolocate myself, I'm speaking from Bari, that is in Puglia, a region called the Hill of Italy in the south. Here you can find good food, good weather, Caribbean Sea, and of course, good science. So I really invite you to visit this wonderful region of Italy. Back to science. So our project is related to the construction of artificial simplified autotrophic protocells with a semi-synthetic bottom-up approach. This architecture must be capable to transduce light energy in chemical energy stored as ATP molecules. We used two different approaches, a single compartment approach and a multi-compartment approach. In the first one, the building blocks are single protein complexes extracted from living organisms and then reconstituting the bilayer of an artificial compartment. In the second one uses fragments of biological membranes that can act as photosynthetic organelles within the artificial compartment. The organism we choose from which extract these building blocks is the purple bacterium Rhodobacter sphaeroides. These bacteria, in particular grow conditions, form spontaneously membrane invaginations called ICM, intracytoplasmic membranes, that contain their entire photosynthetic apparatus. Photosynthetic machinery consists of three protein complexes: the reaction center, RC, the BC1 complex, and the ATP synthase. Under continuous irradiation, reaction center and BC1 complex are coupled to produce a redox photocycle that generates a proton gradient across the cytoplasmic membrane. This proton gradient is then exploited by the ATP synthase to synthesize ATP from ADP and phosphate. For the single compartment approach, we focus the attention on the reaction center protein that is the first protein complex involved in the transduction process. The RC has been extracted, purified, and labeled with the fluorescent antenna. The red ring in the confocal image confirms the RC reconstitution in the lipid bilayer of the giant vesicle. A green hydrophilic dye has been encapsulated to demonstrate that giant vesicles were sealed and no leakage occurred. With an orientation assay, we could prove an alignment of the protein of 90% with the physiological orientation. It means that the reaction center is capable to translocate protons from the internal volume of the vesicle to the external environment. This was confirmed in these confocal images in which, under continuous near-infrared illumination, the grid fluorescence increases inside the giant vesicles due to the presence of the pH-sensitive dye pyranine that increases its fluorescence as the pH increases. With quantitative image analysis, we could calculate an internal pH increase of 0.061 pH units per minute. Moving to the multi-compartment approach, when the cell is broken, several processes occur. The preformed vesicles are released. The intracytoplasmic membrane invaginations pinch off, while the tubular vesicles fragment and seal, producing the bacterial vesicles called chromatophores. These nanometric compartments generally have the photophosphorylation machinery in their membrane with an inside-out orientation. It means with the F1 domain of the ATP synthesis pointing outward, and also encapsulate cytochrome C2 in the reduced form in their internal volume. In this way, they can produce ATP under continuous illumination externally, so they can act as photosynthetic organelles for artificial compartments, such as giant vesicles. Obtain a high yield of chromatophores with the desired orientation of the photophosphorylation machinery, we have employed an extraction procedure based on direct cell lysis by a single French press passage, followed by a first centrifugation in order to remove the cell debris from the sample, and then the solution has been ultracentrifuged to obtain chromatophores as pellet. Chromatophores give this spectrum with the characteristic peak at 860 nanometer for the R26 mutant strain of the Rhodobacter Sphaeroides bacteria that has been used for this study. This spectrum also shows this spectrum lacked the peaks of the carotenoid and the light harvesting complexes too. Chromatophore diameter was measured by dynamic light scattering, giving a mean value of 80 nanometers, while their zeta potential was minus 30.9 millivolt, as expected for the presence of roughly 60% of anionic phospholipids in the Rhodobacter sphaeroides membrane. Cryo-electron microscopy and cryo-electron tomography confirmed the presence of closed spheroidal chromatophores, although some elongated vesicles and open fragments are also present in the sample. Direct inspection of cryo-EM images revealed the presence of the F1 domain of the ATP synthase in the functional orientation on the membrane of closed chromatophores. On average, we found 1.6 ATP synthase per chromatophore, all being outward oriented and thus able to produce ATP in the external solution. In no cases, inward oriented ATP synthase were found. The ATP synthase surface density can be estimated combining DLS and cryo-EM analysis, obtaining 80 ATP synthase per micron squared. In order to be functional, chromatophores with an inside-out morphology should also contain a sufficient amount of cytochrome C2 in their lumen, in the reduced form. We have determined the ratio between functional chromatophores and all other types of non-functional particles and fragments by a light flash excitation experiments. In this assay, the RC spectral change at 860 nanometer due to a photo-induced charge-separated state is recorded immediately after the flash. From the difference between the amplitudes obtained before and after the addition of the detergent to the sample, we determined that 70% of the total amount of the chromatophores are in the functional state. It means with the F1 domain of the ATP synthase facing outwards and containing a sufficient amount of cytochrome C2 in the reduced form. Assayed also chromatophores in bulk for their capacity to produce ATP in the external medium upon continuous illumination in the presence of externally added ADP and phosphate. The amount of synthesized ATP after 3 minutes of near-infrared light irradiation was quantified by the luciferin-luciferase bioluminescence assay according to a calibration line. The ADP to ATP conversion is high but not completed, around 60%, independently to the amount of the ADP added. The measured production of 1.2 millimolar ATP in 3 minutes corresponds to an estimated ATP synthase turnover number of 80 seconds to the -1. That is in good agreement with the highest reported value in literature that are 50 and 90 seconds to the -1. After that, we tried to encapsulate chromatophores within giant vesicles using the droplet transfer method as preparation method. Preliminarily, chromatophores were labeled with the fluorescent dye Nile red. After that, they were added to internal aqueous solution that has been emulsified in mineral oil suspension and obtained by pipetting this emulsion that is stabilized by the lipids dispersed in a mineral oil solution. Then the emulsion is stratified above biphasic system, and then a centrifugation allows the droplet to cross the interface region and obtain vesicles as a pellet. The confocal images confirmed the well-localized fluorescence due to the labeled chromatophores, again indicating that vesicles were sealed. Protocells prepared with all required components are then assayed for ATP production. In the presence of ADP and phosphate, the photoredox reactions can cycle very efficiently because the pH gradient and the electrochemical potential are continuously generated by light absorption and thereafter dissipated by ATP synthase. The quantification of intraprotocell ATP can be carried out again by the luciferin-luciferase bioluminescence assay after releasing ATP from the protocell interior. Preliminary experiments show that detergent-based lysis interferes with the assay, so ATP was released from protocells and quantified after freezing and thawing the sample. The average concentration of intraprotocell ATP has been calculated with the standard addition method. Having shown that protocell can produce ATP from photon energy, then our photosynthetic protocells were built by including in the protocell lumen ADP, phosphate, a linearized plasmid as DNA template, the enzyme T7 RNA polymerase, and the three nucleotides GTP, UTP, and CTP. This because the ATP must be supplied by our chromatophores. The fluorescent dye acridine orange was added externally to perform vesicles to evidence the RNA produced from the coupled photophosphorylation transcription processes. Acridine orange binds both DNA and RNA, emitting a green light. Since the DNA concentration is constant, the variation of acridine orange fluorescence is only due to the progress of transcription. The accumulation of RNA inside our protocells is clearly detected by the increase of the acridine orange RNA complex fluorescence in the confocal images. On the right is reported the average inner fluorescence versus time, obtained by quantitative image analysis of our protocells micrographs per each time interval. Because anti-ATP is the limiting reagent for T7 RNA polymerase, the transcription process ends after 90 minutes, indicating that all ADP has been converted in ATP. Okay, just to summarize what I showed here in this talk, we have seen that a hybrid multi-compartment system can be assembled using giant vesicles as artificial compartment and chromatophore as biological photosynthetic organelles. Chromatophores are the simplest ready-to-use ATP-producing building blocks and can be employed in a modular design of artificial protocells, since we have seen that the encapsulation of chromatophores in giant vesicles with the droplet transfer method is very easy and they retain their photoactivity. The ATP photo-produced can sustain the DNA transcription process within the hybrid multi-compartment protocells, and I hope I convinced you that this hybrid design can be further expanded to include other easy-to-extract natural organelles, increasing the versatility of bottom-up approaches for the construction of artificial protocells. So I would like to thank all the collaborators of this work, starting from Fabio Mabelli and Paola Albanese from my group in Bari, Pasquale Stano at the University of Salento, Massimo Trotta and Francesco Milano at the CNR of Bari, Roberto Marotta at the IT of Genova, and Michele Fiore of the University of Lyon. And of course, I would like to thank you for your kind attention.
Great. Thank you so much for that excellent talk, Dr. Altamura. So I'll go ahead and start with a question. I'm just wondering about, so you show that you isolate the chromatophores on your own, and if there would be some scalability to this process. So for example, if you were talking about building a solar cell from these types of architectures, do you think that that would be a feasible extension of your work?
Can you repeat the question, sorry?
Yeah, so I'm wondering if you think that in terms of scalability, could you actually purify enough material in order to implement some of your architectures into a solar cell for an idea of an application?
Yeah, the process is scalable because I mean, this extraction for my purpose starts from 1 liter of culture of bacteria. So this is enough to prepare my samples. But of course, with the bioreactor, you can produce tons of chromatophores because it's a quite easy process to extract them. And the point is that there's the advantage that in this way, all the photosynthetic machinery is in the physiological environment. So the yield is the highest as possible outside the living beings. But of course, the stability is not so long to create a device as they are, just extracted from bacteria. The other point is that for this study, we used the R26 mutant strain that is not the best choice in efficiency, but is the best choice for to avoid any spectral interference, because this is, I mean, Rhodobacter sphaeroides is a purple bacteria, but as you have seen in the bottle, the bacteria appear green. So this is why we used all the dye in the green region, because we had this spectral window that was helpful for this purpose. But already with the wild type strain, the efficiency increases a lot. And actually, at the moment, we are working with that. So I will give you a more precise answer in the future.
Okay, great. We'll look forward to it. Okay, we have a question from Gabriel Lopez. He says, beautiful work. Is there a means of controlling the size of the lipid sponges? Your last slides implied an oh, I'm sorry, that was from before. Still from him though. For how long can the chromatophores continue to produce ATP? And what ultimately limits the continued generation of ATP by chromatophores?
Okay, so in this moment, of course, the limiting reagent is what we supply. So we have everything from the commercial kit for the transcription that was in excess, and just the ATP was added by us. For just ATP production, now there is not a cycle in order to hydrolyze ATP to obtain again ADP. So it's not a cycle. So it's just one-way reaction, and there is not another reaction that consumes ATP. So we can continuously monitor the ATP producing of chromatophores. But just for stability, I can tell you that they continue to be photoactive for 4 days, stored at 4 degrees. This is the best condition we found right now.
Okay, great. Well, thank you very much for sharing your work with us today. We greatly appreciate it. Okay, and then we'll just end today with a few more reminders. Just that next week we have 2 more speakers on May 17th leading into our virtual conference. So we have next week Professor Joachim Spatz and Dr. Ilya Platzman, both from the Max Planck Institute for Medical Research. And that is all for today. So we thank everyone for your attention, and we'll see you next time around. Thank you.